Aerobic life depends on a paradox. Organisms rely on mitochondrial oxidative phosphorylation to generate the ATP required for survival, yet this respiratory process produces reactive oxygen species, or ROS, as obligate toxic byproducts. Unmitigated oxidative stress causes macromolecular damage. When highly reactive ROS accumulate, they begin stripping electrons from structural lipids, functional proteins, and genomic DNA, acting as a pathological driver of aging and neurodegeneration. To counter this persistent threat, eukaryotic cells evolved a multi-tiered cytoprotective defense mechanism. At the center of this network is the NFE2L2 gene and its protein product, NRF2. Global transcriptomic analyses identify NRF2 as a massive regulatory node. It governs the expression of over 2,000 downstream target genes, integrating cellular redox status with intermediary metabolism, mitochondrial biogenesis, and DNA repair. A cell's ability to manage physiological stressors depends on the fluid tuning of this axis. This allows for specific adjustments to transcriptional activity based on the precise nature of the metabolic or chemical threat. The regulatory precision of NRF2 is dictated by its structural topology. The protein sequence is divided into 7 conserved NRF2 ECH homology domains, designated NEH1 through NEH7, which facilitate the protein-protein interactions required for transcriptional control. Under homeostatic conditions, NRF2 is kept on a tight leash, maintaining a biological half-life of roughly 15 to 40 minutes. This rapid molecular turnover is exclusively dictated by the KEAP1 protein, the primary cytosolic repressor of NRF2. KEAP1 physically controls NRF2 through a sequence known as the hinge and latch model. One KEAP1 monomer binds to the high-affinity ETGE motif on the NET2 domain, acting as a permanent hinge. Simultaneously, the second KEAP1 monomer binds to the adjacent low-affinity DLG motif, acting as a dynamic latch. Once NRF2 is tethered in this specific conformation, the associated COL3 RBX1 E3 ubiquitin ligase complex polyubiquinates adjacent lysine residues. This flags the transcription factor for destruction by the 26S proteasome. KEAP1 functions as the cellular redox sensor through a complex cysteine code. The protein is populated with reactive cysteines, specifically cis-151, cis-273, and cis-288, which are susceptible to chemical modification by electrophiles. When reactive oxygen species or exogenous electrophiles react with these critical cysteines, the modification induces a conformational shift within the KEAP1 dimer. This change structurally disrupts the low-affinity DLG latch, preventing the alignment required for NRF2 ubiquination. This mechanical failure of the KEAP latch stalls the degradation machinery. As a result, newly synthesized NRF2 bypasses the disabled repressor complex, accumulates in the cytosol, and undergoes immediate nuclear translocation. Canonical KEAP1 sensing is not the only way the cell manages this axis. Alternative pathways exist that integrate NRF2 signaling with autophagic and metabolic stressors. One prominent non-canonical pathway involves a feedback loop between NRF2 and the autophagy adapter protein P62, also known as sequestosome 1. The P62 protein contains a KEAP1 interacting region that harbors an ETGE-like motif. Structurally, it is virtually identical to the high-affinity hinge motif found on NRF2 itself. During periods of autophagic flux, P62 accumulates in the cytosol and outcompetes NRF2 for binding to the KEAP1 repressor. By physically sequestering KEAP1, P62 stabilizes NRF2 in a manner that is independent of both cysteine modification and the cellular redox state. To terminate the response once homeostasis returns, the cell utilizes the NES6 degron as an off switch. Active GSK3-beta phosphorylates specific motifs in this domain, allowing the beta-TRCP ubiquitin ligase to bind and clear NRF2 from the nucleus. These alternative degradation and rescue pathways allow the NRF2 axis to function as a holistic systems manager, responding to total cellular metabolic health rather than localized radical scavenging alone. Upon entering the nucleus, the execution phase begins. NRF2 heterodimerizes with small MAF proteins at the NEH1 domain, allowing the complex to bind to antioxidant response elements, or AREs, located within target gene promoters. The cell immediately deploys its antioxidant defenses. NRF2 upregulates the multi-step biosynthetic pathway for glutathione, alongside bioredoxin systems, phase 2 detoxifying enzymes, and the direct antioxidant protein heme oxygenase 1. NRF2 redirects cellular metabolism to fuel these defenses, shunting carbon flux into the pentose phosphate pathway. This metabolic shift maximizes the production of NADPH, which is strictly required to regenerate reduced glutathione. Repair programs are initiated to address cellular architecture. NRF2 drives proteostasis by regulating the assembly of the 26S proteasome, while directly orchestrating DNA repair through the regulation of OGG1 for base excision repair and BRCA1 for homologous recombination. Beyond activating genes, NRF2 can repress them. It physically binds to the promoter proximal regions of the IL-6 and IL-1 beta genes, structurally obstructing RNA polymerase II to silence the transcription of these pro-inflammatory cytokines. For long-term structural maintenance, NRF2 upregulates nuclear respiratory factor 1, which subsequently transcribes mitochondrial transcription factors to drive biogenesis. The downstream transcriptome does more than neutralize a temporary threat. It rebuilds the metabolic infrastructure and genomic defenses required to survive catastrophic environmental shifts. Mammalian biology utilizes exogenous natural compounds from the diet to modulate this internal signaling network. Most of these NRF2 activators share a specific biochemical trait. They contain alpha-beta unsaturated carbonyl groups. These structural moieties enable the molecules to act as lipophilic mycoacceptors. The benchmark natural activator is sulforaphane, an isothiocyanate found in cruciferous vegetables. Sulforaphane crosses serositolipid bilayers to execute a Michael addition reaction, covalently modifying the KEAP1 C151 residue to unleash NRF2. Sulforaphane provides a two-pronged activation. It also induces DNA demethylation at the NFE2L2 promoter, restoring the baseline expression of NRF2 mRNA. Other modifiers like curcumin and EGCG utilize distinct electrophilic or oxidative modifications to break the KEAP1 latch, triggering the same nuclear translocation and adaptive response. Dietary antioxidants do not provide benefits by passively absorbing free radicals. Instead, they act as mild, targeted stressors that trigger the expansive NRF2 genomic battery. This illustrates the principle of hormesis, the process by which exposure to a low dose of a damaging agent induces an adaptive biological response. Physical exercise is a primary driver of hormesis. Mechanical stress on the plasma membrane activates NOx2, generating a transient burst of ROS and nitric oxide. This oxidatively modifies KEAP1, liberating NRF2 to initiate mitochondrial iogenesis and heightened antioxidant capacity. Thermal therapies like sauna and cryotherapy utilize this mechanism. The transient sympathetic stress and mild cellular insult act as the signaling trigger for NRF2 stabilization. While controlled stressors promote health, unmitigated massive exposure to environmental toxins has the opposite effect. Fine particulate matter from massive fossil fuel combustion triggers uncontrolled ROS generation that forces a desperate survival response. Initially, NRF2 attempts to detoxify these pollutants through phase 2 conjugation. However, chronic exposure to diesel exhaust and chemical carcinogens can overwhelm the buffering capacity, leading to pathway exhaustion and oxidative collapse. NRF2 efficacy operates on a bell curve. Calibrated transient stress builds resilience, but a chronic toxic load forces the system toward failure. The NRF2 axis is an integrated biological router featuring bidirectional crosstalk with the inflammatory NF-kappa B axis, the p53 tumor suppressor, and aryl hydrocarbon receptors. The mammalian cell uses its own degradation machinery to translate chaotic external threats into a coordinated genomic shield. Health is not achieved by attempting to eradicate all biological stress, but by training and tuning the cytoprotective network through targeted exposure. Modulating the NRF2 hinge and latch represents the frontier of translational medicine, offering the capability to rewrite the rules of metabolic disease, neurodegeneration, and the human lifespan.